Hi everyone, nice to meet you. I'm Chang Liu, a former postgraduate student at the University of Cambridge, and I'm currently working at AMD in charge of benchmarking large language models running on AMD GPUs. Today, it is my pleasure to give a relatively comprehensive introduction of our work to you, and um, I'd like to hear about your feedbacks and your follow-up questions about our work in the future. And our work has been just accepted by the workshop Machine Learning in Structural Biology, which is one of the workshops at Europe's 2025. And the title of our work is shown on the slide, Multiscale Protein Structure Modeling with Geometric Graph Units. And all the co-authors and our supervisors are listed on here too. This work was originated from a module assignment during our study of geometric deep learning at the University of Cambridge. Okay, so now let's get started. My presentation today consists of a few parts which are listed on the slide. It includes background, problem to solve, our method, and after that, I'm going to give some experimental results and a brief run through of the theoretical proof of our method. After all that, there comes to the conclusion and discussion section. So let's dive into the background first. Our paper mainly focuses on the task of protein fold classification. So what is protein fold classification? Protein fold classification consists of two terms, protein fold and classification. So now I want to play an interaction game with all the researchers from CIG. I'm wondering whether you have heard about the concept of protein fold or protein fold classification before. If so, you can send a message in our chat box or unmute yourself to say yes.
Yes, yes.
Okay, great. Okay, so the reason why I asked this question is I didn't know much about this task before I took this module at my postgraduate study. So I would give a very brief introduction of this term first. So we can take a look of the picture on the right side. This picture illustrates the hierarchical organization of protein structure. The primary structure is the linear sequence of amino acids, which determines how local secondary structures such as alpha helices and beta sheets form through backbone hydrogen bonding. And then these secondary elements pack together into unique three-dimensional tertiary structure driven by side chain interactions. Finally, quaternary structure arises when multiple folded protein chains assemble into a functional complex. In other words, a protein fold is the topology including connectivity and order of secondary structures in proteins. And it also represents the spatial arrangement of alpha helices and beta sheets and denotes the overall 3D architecture of the protein domain. So protein fold actually is the third part of the diagram on the right. And what is protein fold classification? This task groups proteins into categories based on the overall three-dimensional arrangement of their secondary structure elements. So actually, we're doing classification based on this kind of structure. And then you may ask why we focus on protein fold classification. So from the task itself, protein fold classification is important because it links protein structure to function, evolution, and annotation, especially when sequence similarity alone is insufficient. And from the perspective of our study, we hope we hope our proposed method can be extended or can be validated on not only protein fold classification, but also other relevant tasks like inverse folding. So from what we learned previously is protein fold classification serves as a litmus test for the ability of a model to distinguish different structural folds. It stands to reason that models that perform poorly on distinguishing fold classes likely learn limited or low quality structural representations. And this is quoted from one of the previous papers named Protein Workshop, also was contributed by one of our supervisors, Chatanya's work. So that's why we hope to focus on protein fold classification in our study. And so before locating the issue that we want to solve or we want to improve, we did some research on this area, and we can see a trend shown on the slide that since around 2017 to 2018, there are a lot of approaches based on geometric graph neural networks and transformers emerged and achieving good performance in different applications such as protein fold classification and protein structure prediction, etc. And on this diagram, you can see that a lot of methods based on traditional geometric graph neural networks achieved very good results and improvements. And at the same time, there appeared another type of special kinds of graph neural network named transformer, which has also been leveraged into the pipelines and been proved to be very effective in applications in protein analysis. Taking AlphaFold as an example, which is very famous now and was first proposed also around 2017 to 2018. AlphaFold solved the protein structure prediction at scale, predicting accurate 3D protein structures directly from amino acid sequences rather than depending on extra information from the protein. So you may ask why AlphaFold was so impressive, because it predicts highly accurate 3D protein structures. And also, it effectively solves a over 50-year challenge in biology, and it enables the possibility to accurately predict protein structure at a very large scale. Besides, AlphaFold further proved that an end-to-end differentiable pipeline, like an end-to-end neural network, can achieve very good performance in protein analysis. Apart from all these kind of parts, AlphaFold also boosts the development of its downstream tasks, like a protein fold classification, what we are focusing on in this work, and also functional annotation and drug discovery. So, based on the study of previous works, we can sort of categorize the previous works into two types. They are geometric graph neural networks based methods and transformer based methods. And both of them have become state-of-the-art methods for learning from 3D protein structures. But as I have mentioned, actually, transformer is a special type of graph neural network. It is actually a fully connected graph neural network. Yeah, hello? Okay. Um, okay. So, any questions? Okay. I feel like that might be an issue of the soundtrack. Anyways, so for the transformer itself, it is a fully connected graph neural network. And in the latest thesis of Chaitanya, it's named as transformers are graph neural networks, which also talk about this idea. But in our categories, we just like classify this kind of two types of workflows in this kind of analysis. Because traditional geometric GNNs and transformer-based methods have some difference based on their ways of building the graphs. As I have mentioned, that transformer is a fully connected graph, while geometric GNNs sometimes are not. Okay, so after analyzing and studying the previous work, we can sort of summarize the problem that we want to solve. Because we notice that the traditional geometric GNNs has a reliance of message passing, which prevents them from capturing the hierarchical interactions that govern protein function, such as global domains and long-range allosteric regulation. So on the right side, we illustrate an example of the long-range allosteric regulation. We can see that for allosteric site, there is some distance between its location to the active site of this protein. So if only use the traditional graph neural network, our model would be limited due to its local perceptive field. And on the other hand, transformer-based models would consume significant computational resources because they build this kind of fully connected graph, no matter what kind of protein structure we input into the model. So here comes to our method. In our paper, we argue that the network architecture itself should mirror what we have observed, that the geometric GNNs should have the capability to capture longer range information contained in the protein structure. So we introduce geometric graph UNets, a new class of models that learn multi-scale representations by recursively coarsening and refining the protein graph in our work. And before designing this framework, we studied a few works in hierarchical geometric GNNs, which can be categorized into three types. They are local pooling, virtual nodes, virtual edges. So for local pooling, it clusters nodes at each GNN layer to form new nodes for the next layer, with approaches ranging from basic clustering to learnable and attention-based adaptive pooling. So our research actually extends the benefits of pooling via a UNet architecture, which are shown on the right side of the slide. Of which this kind of UNet architecture, the effectiveness has been demonstrated in private studies in point cloud data processing. So let's take a detailed look of our overall pipeline of our method. So this is the overview pipeline of the whole pipeline of our method, and we mainly followed the pipeline of the protein workshop, the 38 reference paper in our references information. And you can see there are mainly 5 phases of this pipeline. The first one is protein structure datasets. So if you want to train any neural network, you need to find the right dataset. For our case, we downloaded the open and free dataset named Scope from the internet. And if you're curious and interested, you can also try our models with some other datasets like CAS, which is also a very well established dataset in protein structure. Besides, For a specific scenario in applications, researchers and scientists can collect corresponding data sets according to your real demands. So with this kind of protein structure data set, we can apply the featureization to convert those data into a format where our GNN models can process and can understand. So as mentioned before, we follow the main steps of protein workshop. We also adapt a manual designed featurizer in our method. For now, researchers tend to leverage a pre-trained protein language model to generate the features, which has been proved to be more effective to capture those features of the protein structure. But in our method, we didn't try any protein language models. We only leveraged the previous manual designed featurizers. And after got this kind of features of the proteins, we send them into our well-designed GNN models, which is our hierarchical GNN. And I will give a very detailed introduction of the architecture of the model later. And after this part of after processing and representing the protein structure using GNN layers and GNN models, we would attach an output head, which is not shown on the slide, but it is also it is all usually an MLP decoder, which would convert the graph embedding learned by the GNN models to a target output, like a target fold classification number, or the number hinting the expressivity of the input protein structure, or the models, or the graph neural networks. So our novelty is actually implemented in the GNN models phase. Okay, so let's take a detailed look of our model architecture. And you can see it is a U-net shape model, which consists of several stages or several blocks of pooling and unpooling phases. And from this kind of a diagram, you can see that it consists of like a encoding phase and a decoding phase. So as we have mentioned before, a protein function is governed by a structural hierarchy. Standard GNNs struggle to capture these long-range interactions because of their limited local perceptive field. And our proposed geometric graph UNet is a biologically inspired architecture that encodes a protein graph by progressive coarsening, moving from local representations to global ones, which would enhance the capability of the model itself to capture long-range features and and the informations from the protein structure. So, in order to build up this kind of U-net shape, we introduced two types of pooling layers in our model architecture. And this slide demonstrates the difference between the two types of pooling layers we introduced. They are point pooling layers and the sparse pooling layers. So the pooling layers help us connect the distance nodes and meanwhile enlarge the receptive field. Why is that? Is because they sampled a subgroup of nodes as supernodes first, and then they would they would aggregate all the information from the original nodes of the supernode to the supernodes in order to sort of enlarge the scale of the perceptive field and meanwhile reduce the information or the node numbers in the graph. And we can see that it saves some computational resources compared to transformer models. So let's take a detailed look of the definition of point pooling layer and sparse pooling layer. So for point pooling layer, it was used for point cloud data in PointNet++ before. So in our work, we transferred its capability point cloud data to protein structures, which have been proved be effective in our experiments. And let's take a specific definition. I'll take a specific look of its definition. So for each pair of nodes, Vi does not belong to Vp. And Vp is like the sampled supernode set of the next graph neural network, of the next graph neural layer. And the supernodes V, S, and J, which stands for V supernode, the J's supernode, where Vi denotes the ith node, Vp stands for the sampled node set, and V superscript supernode, subscript J, is the J supernode. We define the cluster assignment matrix C as C subscript Ji equal to 1 when the ith node belongs to the neighborhood of the jth supernode. Otherwise, it's going to be set as 0. So from this kind of definition, we can see that point pooling layers would aggregate some node information for multiple times, because for some supernodes, they may have some overlapped neighbors, as you can see from this illustration clearly. For the node on the right, its neighbor is like the 4 original nodes. But for the like left node, its neighbor was the other 4 nodes, and you can see they have 3 nodes as the overlapped nodes in together. And with this kind of definition, it might reduce some capabilities of the graph representation, which we would demonstrate and discuss more about it in our experiment section. Okay, and for the aggregation of the features of the graph node, we leverage the message passing layer from Jing to update the features of each pooled supernode. What's worth mentioning is we applied a summation operation to aggregate these features, and we also tried with some other operations like concatenations, but we found out summation is more effective compared to concatenation, and that's the reason why we leverage the summation in this setting.
Hey, Chang,
Yeah.
um, on the on the pooling point layer where we talk about the supernodes, I think the previous slide,
Mm-hmm.
it's an exciting concept, and I'm trying to understand. We have
Mm-hmm.
Yes, we have this assignment matrix, like sort of being in the cluster, but also the concept of supernodes. How do the two link together? Like, how do we form the supernodes, but also how do we come up with this assignment cluster?
Okay, it's a very good question. So for the supernodes, we applied a sampling algorithm to to sample a subset of nodes from the original node set. For example, V, right? So V is like the original set of the nodes in the first layer of the model. And we applied a sampling algorithm to select a subset of nodes and
Okay,
form them as a supernode. Yeah. Actually, they are just like sampled randomly
so this
according.
sampling,
Yeah.
the sampling happens at a global scale, not in within local maybe nodes within a given sort of segment, but the entire protein um, chain, like you sample globally.
Yeah. So in this kind of case, we leverage the sampling algorithm named as the FPS, like the farthest point sampling. So
Oh,
this kind of algorithm aims to sample as average as possible of the whole graph. But from your like question, we can also extend the sampling method to other strategies. Like maybe you can sample it according to the neighborhood, to the importance of the node, etc. But now we just use this kind of FPS sampling algorithm, following the protein workshop as well. Yeah.
okay. So now the cluster
Mm-hmm.
assignment matrix. See, how does it come into the link?
Say again.
The cluster assignment matrix see, that is being defined here.
Yes.
What is its connection with the supernodes and how does it come
Oh,
into the link? Yes.
okay, I see. Oh, very good question. Very good question. Um, so the new edges would be generated according to the new according to the new graph, like according to the new supernode set.
Okay.
Yes, I need to like investigate onto like our paper for the detail, but it can be like those edges can be generated according to the proximity of the supernode, and or it can be generated according to other algorithms.
Okay. Okay. Sure.
Mm. It's a very good question, Lookman.
Thank you. Thank you.
Okay, do you have any other questions?
No, no.
Okay, cool. So let's dive into the sparse pooling layer. So for sparse pooling layer, um, it's it is similar to point pooling layer, but what's different is it partitions the structure into distinct, non-overlapping regions, which means for sparse pooling layers, we only assign each node to one supernode. So it reduces the overlapping of the subsets we have mentioned before in the point pooling layer. And you can see from the definition here, the major difference is the rule to determine whether C subscript JI can be set up as one is decided by like the nearest neighbor of the supernode. So each node can only be assigned once when it's nearest to the supernode. And for the reduction function, they leverage the same mechanism and operations we have mentioned in the point pooling layer. Okay, so after introducing the two types of pointer polling layer and the sparse polling layer, now here comes to the section of defining the overall module architecture of the geometric UNet. Actually, we also follow as mentioned before, we follow the main pipeline of the protein workshop. So we mainly implement the newly introduced UNet architecture into the into the encoder phase of protein workshop. And this UNet architecture comprises of an encoder and a decoder path with pooling and unpooling blocks. The unpooling block actually integrates pooled and unpooled features through an unpooling layer. The unpooled graph, you can see that, like is the path on the right side. So the unpooled graph is reconstructed with a skip connection concatenating cache the features from the corresponding polling blocks to the unpolling output. So this is just like one of the most important design in our UNet work, in our UNet arc. So what's more specifically is we initialize all the scalar and vector features to zeros in the reconstructed unpolling features. And why this kind of skip connection is very important is because along with the unpolling phase of the UNet, we sort of reconstruct the graph of the protein fold, of the protein structure, right? And we integrate the previous features from the pooled blocks to the unpooling blocks. This kind of set sort of integrate the features from different layers or different scales of perceptive fields of the protein structure. So that is like the key design of our model. And at the same time, we didn't introduce much extra parameters in our model because for all those unpooling blocks, we only set up like a very simple layers to integrate those pulled and unpulled feature together. But we didn't introduce any extra graph networks like graph layers. So it's very computational effective as well. Any questions about this?
Yes, yes,
Okay.
Chang. There was a
Yeah.
question in the chat. Maybe I should read it.
Mhm.
Gabriel is asking what happens if too much detail of information is compressed into a single supernode. I think in that pooling process is wondering if maybe you form a supernode with a lot of information. So I could repeat the question. What happens if too much detail of information is compressed into a single supernode? Is,
Okay.
is that a possibility anyway?
Yeah, it is.
Okay.
It is possible. Um, okay. Let me think of it. So, um, if it happens, I think the supernode will become very important.
Mm.
And from our strategy of sampling, we only leverage the farthest sampling strategy. So we would not consider much about the importance of the node itself in the beginning. I mean, by default. So maybe the supernode with much importance might be sampled out, like might not be sampled to the next layer. That's potentially possible based on our sampling algorithm. But you can also apply a different strategy of the sampling. You can add up some importance weights into the sampling algorithm to have the graph maintain the most important nodes as first. Yes, Benjamin.
I think Benjamin, were you saying something? I
Oh, okay, it's fine.
okay.
Okay, cool. So okay, that's just like the basic definition. Um, okay,
Yes.
Lukman, you have a question about the.
Yeah, I think what I wanted to ask in line with that question is as the pooling is performed, and my intuition here is geometric pooling here that is happening. And you will correct me if I'm wrong, that we need to preserve the 3D structure as we are performing the pooling of the protein molecule that is forming this graph structure, the 3D. So is the possibility of the sampling algorithm forming uh, a supernode with much information or with detailed information in that case. Wouldn't that lead to collapse of the 3D structure in the pulled graph if that happens?
Oh, okay. It is a very good question too. So the 3D information have been embedded or have been featurized in our graph network by the featurizer I have mentioned before, right?
All right.
So, so what we can see now is actually an abstract graph where
Oh,
all the 3D information from the protein actually have been embedded into each node. So, um, so maybe your concerns would like possibly happen. Maybe some important information would be sampled away. But I think like leading to collapse, it would not be like a
it's an extreme event.
yeah, it's a very extreme
Mm.
case, I think.
I
It
see.
will
I
not
think
happen
you've
very
you
frequently.
have nailed it. You have nailed it for me.
Okay, thank you.
Thank you.
Okay, so okay, so after introducing the method, let's dive into the experimental results of our paper. We actually conducted two sets of experiments. They are fold classification, and the other one is synthetic expressivity using K-chains. So for protein classification, we have mentioned before that it is very important to to see whether a model can distinguish different structural folds, and it can also test the capability of the proposed methods of whether they can extend whether they can be extended to other tasks. So we also follow the settings of Protein Workshop. We leverage the dataset, the scope 1.75, and downloaded them from the internet. We trained all the model using PyTorch and a single NVIDIA RTX 4090 GPU. And for the training settings, we set up to 150 epochs using Atom Optimizer and learning rates ranging from 0.001 to 0.01. And we demonstrated two metrics, accuracy and F1 score, in our paper. But actually, while we were conducting the experiments, we also have F1 max score, which is similar to the protein workshop. So on this slide, we can see the experimental result. The results are reported on three test subsets. They are fold, superfamily, family, which classify proteins into 1,195 folds. And the numbers in accuracy and F1 score are averaged through three random runs. What's worth mentioning is the baselines, SheNet and GVP, are also tuned with decoders with fewer hidden layers with higher performance. So the decoders here is not the decoder we have mentioned in our UNet. It's the decoder of the whole pipeline of the protein workshop that we have illustrated in previous slides. So the decoder is actually the output head. And in our experiment, we sort of first tuned the baseline's performance. So the numbers of SheNet and GVP here are higher than their numbers on the protein workshop paper or even their original paper. And from the table here, we can see UNet point pool and UNet sparse pool. They respectively stand for our UNet variant model with point pooling layers and sparse point pooling layers introduced. And you can see that overall, the UNet sparse poor variant would outperform all the other models, which can achieve the overall best performance. And um, and I have to enhance one more thing, is emphasize one more thing, is that for a fair comparison, we also reduce the parameters in our variance so as to make sure that our variance of our UNet models maintain exactly the same or less parameters than the baselines, like than the SheNet or GVP. So from the table, the params column, you can see that like for the SheNet, for the tuned SheNet, it is there is 1.23 million parameters. And for our UNet variants, there are some extra parameters involved in our UNet architecture, but we reduced some parameters from the decoders so as to maintain the fairness of the comparison. And for GVP variants, we have less parameters compared to the baseline. So you can see if we introduce extra UNet architecture based on the baseline itself, we can actually outperform the baselines more, like more than the numbers we showed in this table. But for a more fair comparison, we did this tuning of the like the baseline in our variants as well, which also showed the effectiveness of the UNet architecture. Okay, so here comes to the second experiment we had in our paper. It is synthetic expressivity using K-chains, which was proposed by Chaitanya. And this experiment aims to exploring how different numbers of pooling layers would enhance expressivity on synthetic K-chain graphs. And for a K-chain graph, we can see the illustration on the right side. Remove the first and the last node of the chain. There would be K nodes kept in the chain, and that is the K-chain definition. And from the study of geometric Westphaler-Lyman's test, it requires the floor of k over 2 plus 1 iterations to differentiate k-chains. So through this kind of experiment, we want to see whether first, all models can distinguish k-chains with fewer layers than the minimal requirement. And second, we want to see whether the pooling can enhance expressivity of the graph model. Okay, so here is the result. So in this table, each pair of k-chains consists of k plus 2 nodes, with k nodes arranged in a line and differentiated by the orientation of the two endpoints. And GW test requires less than the floor of k over 2 plus 1 iterations to differentiate the k-chains graphs. Therefore, it is theoretically expressive. In this table, we report averages over 10 runs for k equals to 4 and assess polling effectiveness on SheNet, SphereNet, GVP, GNN, etc. And the GWL test requires the this number of iterations to differentiate k-chains. And you can see from this table that we got the answers of the previous two questions. They are both yes.
Chang,
So with yeah,
a quick
recommend.
one. In the context of this table, what does the K chains mean? Like, are we referring to a chain of the proteins? Like,
Oh, it is just like an abstract representation of the graph. Like
okay,
it's just some nodes.
nodes?
Yeah, yeah, some nodes, but it's
Oh,
not corresponding to any protein structure or etc. It's just like an abstract symbol.
okay, okay, got it.
Okay. So from this table, we can see that pooling can enhance expressivity by enabling the model to distinguish geometric graphs it previously could not. Because you can see that the number of layers needed to distinguish the graphs reduced compared to the initial graph neural network definition. Okay, so overall, it is the experimental result in our paper. And for the theoretical proof, please refer to our paper for more details. So what I would introduce here is pool operation introduced in our model can maintain expressivity from the theoretical proof. And for the theoretical proof, we first study expressivity through the lens of distinguishing non-isomorphic geometric graphs, adopting the GWL test. Then we extend the hierarchical expressivity framework introduced for pooling to the geometric setting. And for more details, please refer to our original paper. And if you have any questions, please just send them to us. And I think my co-authors can also be very interested about your feedback and questions about this part too. Okay, so here comes to the last part of my introduction. This is the conclusion of my presentation. First, we observe that geometric GNNs should reflect the hierarchical nature of the biological structures in proteins. So we introduce our geometric graph unit, which can serve as a flexible blueprint for new generation of multi-scale models. So you can also extend or adapt our unit to any other protein fold classification method or protein analysis method, because our design of this kind of unit architecture is more like a blueprint. And last but not least, by treating proteins as nested structural assemblies, we show how multi-scale architectures can yield more powerful representations for predicting function from structure. And for the discussion part, we really welcome contributions from the community and the CIG researchers and scientists. We feel like in the future, we can also extend the validation of our method on a broader range of structure function prediction tasks, like inverse folding. And also, we can apply the idea to model biomolecular complexes with hierarchical interactions across multiple scales. And also, we believe geometric units may be especially effective as encoders for multi-state inverse design pipelines. So I really do hope to hear about a lot of feedbacks and thoughts about our work. And that's the end of my introduction. Thank you.
Thank you, Chang. It has been really an in-depth technical talk and very exciting. Without being unfair, Gabriel had a question, I think, sort of follow up. He was asking if some information is lost during compression process in the supernode, how efficient or effective will the pooling process results be after classification?
Oh, it's a really, really good question. So actually, for the compression of the graph now, I have mentioned that we are leveraging a method named FPS sampling strategy. So in this kind of sampling strategy, we set up a ratio of the sampling. It is set as 0.6, which would cause some information loss. And if which might cause some information loss, but we have to conduct different sets of experiments, and you can tune this kind of sampling ratio with larger number like 0.7, which would keep more nodes as the supernodes in the next layer. So from my point of view is you can tune this hyperparameter in the sampling strategy we're using now. And also you can try to use different sampling strategies to compress the graph so that maybe the results can show that, oh, okay, with this kind of sampling rate, the performance of the model will go up or not. But from our perspective now, we just set up this kind of sampling ratio as 0.6. We didn't try many other settings to compare the different performance. Yeah. So that's it.
Okay. I don't know whether someone has a follow-up question on that, specifically Gabriel. But for me, I think my question would be about the task of protein fold declassification and it being a good test for global structure understanding.
Yeah.
So how does the protein fold declassification becomes a good is it the choice of the understanding of the global structure?
Oh, it's a very good question too.
Yes.
So as I've mentioned on the first few slides before, why we take protein fold classification as our task is because after analysis on protein structure and relevant methods, um, we found out that it's not our findings, it's actually our supervisor's findings. So they found out that models that perform poorly on distinguishing fold classes likely learn limited or low quality structural representations. I think in the number 38 protein workshop paper, they explain about this idea more thoroughly. And
Yeah.
Lookman, maybe you can look up online for the paper for more details about this part.
Sure, sure,
Yeah.
sure. I have to. Um, um, if there are no other questions from the audience, I want to thank Chang for saving up her time and really in this New Year holiday to talk to us. It is really an honor. Thank you very much. And we really thank you. And to the team, to Chaitanya and the team in your lab. Really, thank you very much for really taking this call and providing the call. It is an honor for us. I want to thank you and thank you very much indeed. And to your
Oh,
team, please
thank
deliver
you.